Welcome, everyone. I'm absolutely thrilled to have you here for this explainer. As your senior professor of microbiology, my goal for all of you today, whether you're future MBBS doctors, nursing professionals, BDS dentists, or allied health science experts, is pretty simple. We're going to take those dry, textbook facts about bacterial structure, and we're going to transform them into high-yield, life-saving clinical knowledge. Because you see, understanding the enemy, that's the absolute first step in defeating it.
So, here's our syllabus for today. We're going to build a complete profile of these microscopic adversaries. First, we'll meet the prokaryotes, then look at shapes, sizes, and stains, dissect the anatomy of a bacterium, jump into physiology and bacterial growth, and wrap it all up with some crucial clinical correlations.
All right, let's dive right in. Section 1, meet the prokaryote, the fundamental biological divide. Okay, so here's our biological battlefield. On one side, our target, prokaryotes, the bacteria. On the other side, our patients, eukaryotic human cells. To treat infections without harming our patients, we really have to exploit the differences between these two. Notice how bacteria have a nucleoid with absolutely no surrounding membrane, just a single circular chromosome. They divide by simple binary fission. They've got these smaller 70S ribosomes and, this is critical, a peptidoglycan cell wall. Human cells completely lack that wall. And here is a massive, high-yield fact for your exams, so listen up. Bacteria do not have mitochondria. No way. Instead, they literally have to use their own plasma membrane to house the electron transport chain for respiration. Keep that in the back of your mind, because these unique bacterial structures are exactly what our antibiotics are designed to attack.
Moving right along to Section 2, shapes, sizes, and stains. Identifying the adversary in the lab.
Just look at that number. 2 to 5 micrometers. That's the typical length of medically important bacteria. To put that into perspective for you, a single human red blood cell is about 7 to 8 micrometers wide. So these adversaries are incredibly tiny. And that's exactly why our staining techniques need to be absolutely precise.
You know, physical form is our very first clue in profiling a pathogen. We've got a few primary shapes to look out for. Cocci. Those are your berry-shaped spheres. Bacilli are rod-shaped or cylindrical. Then we have vibrios, which look just like little curved commas. Spirilla are these rigid spiral forms, while spirochetes are slender, highly flexible spirals. Think of the bug that causes syphilis. Actinomycetes almost look like branching fungi. And pleomorphic bacteria, well, those are our shapeshifters. They vary considerably in size and shape depending on their environment.
But here's the thing. Bacteria rarely travel completely alone. The plane of cellular division actually dictates how they group together. So when they divide and stick in pairs, we get diplococci, like pneumococcus. If they form long chains, that's streptococci. When they divide in multiple planes, they form these tight grape-like clusters. That's your classic staphylococci. We even see these complex Chinese letter or cuneiform V-shapes, which is a total hallmark of Corynebacterium diphtheria. When you're looking down that microscope on your clinical rotations, these arrangements are an absolute dead giveaway.
So, how do we see them so clearly? Well, we use Christian Graham's brilliant 1884 technique. It's basically a 4-step recipe. First, we apply the primary stain, crystal violet. Next, iodine acts as a mordant to really lock that color in. Step 3 is the critical decolorizer, using alcohol or acetone. And finally, we apply a counterstain, like safranin. Bacteria that hold on to that initial violet dye through the decolorizing step, those are our gram-positives, and they show up purple. The ones that lose it and take up the pink counterstain are our gram-negatives. It's a seriously elegant protocol that just immediately splits the bacterial world right in half for us.
Okay, let's jump to Section 3, Anatomy of a Bacterium. We're inspecting the pathogen's armor and weapons here.
Now we're shifting focus from how they look to how they're built. I want you to use this mental visualization when you think of bacterial anatomy. Imagine an outermost sugar coat. That's the capsule. It acts like an invisibility cloak, totally preventing our white blood cells from engulfing them. Beneath that is a rigid brick wall, the cell wall, giving it structural integrity. Then you've got a flexible skin, which is the plasma membrane. And finally, inside the whole shebang is the soup, the cytoplasm, just floating with ribosomes and a tangled thread of haploid DNA. Getting a solid grip on this layout is absolutely essential for pharmacology because our drugs are designed to target these very specific layers.
And this perfectly explains the gram stain we literally just talked about. Look at the gram-positive wall on the left. It's got this massively thick peptidoglycan layer, heavily reinforced with teichoic acids. That thick wall is exactly what traps that purple crystal violet dye. Now contrast that with the gram-negative on the right. It has a super thin peptidoglycan layer, but it possesses this sneaky complex outer membrane. And embedded right in that outer membrane is something called lipopolysaccharide, or LPS.
All right, everyone, pens down for a second. Let's do a Professor's Viva. I want you to imagine you're sitting in your final oral exams. The examiner leans in, looks you in the eye, and asks, what is the endotoxin of gram-negative bacteria? Think about what we just saw.
The definitive answer? It's the lipid A portion of the lipopolysaccharide. You absolutely must know this. When a gram-negative bacterium dies and breaks apart in a patient's bloodstream, it releases this lipid A. And this specific molecular component is what triggers a massive immune response, leading to severe clinical reactions like fever and endotoxic shock. It's literally a molecular landmine.
Let's look a bit deeper inside at the internal machinery and surface tools. Those 70S ribosomes, they synthesize proteins. And because human cells have larger 80S ribosomes, we can use drugs like tetracyclines to safely jam up the bacterial machinery without harming the patient at all. Then we have mesosomes, which act kind of like makeshift mitochondria holding respiratory enzymes. We've got plasmids, which are these extra little circles of DNA, and our plasmids in particular drive some terrifying antibiotic resistance. On the surface, pili help them stick to human tissues or even swap DNA, while thread-like flagella act like little outboard motors for motility. See, every single piece of anatomy here translates directly to clinical pathogenesis.
That brings us nicely to Section 4, Physiology and Bacterial Growth, Survival Tactics and Replication.
We know how they're armed, so let's talk about how these pathogens breathe, feed, and multiply inside a host. The most crucial environmental factor here is oxygen, and knowing a pathogen's oxygen preference isn't just fun trivia, it's critical for how you're going to collect and culture samples on the wards. Obligate aerobes like Pseudomonas, they absolutely need oxygen to survive. Obligate anaerobes like Clostridium are actually killed by it. If you suspect a Clostridium infection, you must transport that swab in an oxygen-free container. Facultative anaerobes like E. coli are a bit more versatile. They thrive with or without it. And capnophiles like Haemophilus influenzae, well, they crave just a little extra carbon dioxide to really get growing.
Now, when these bacteria enter a host or a culture tube, they follow this exact growth curve. First up, the lag phase. This is where they're just sizing up the environment, getting ready. Then, the log phase. Listen to me closely here. During this exponential log phase, the bacteria are replicating at their absolute maximum rate. Because they're rapidly building new cell walls and synthesizing DNA, this is the strategic window when they are most vulnerable to our antibiotic weapons. It's a real game changer to know this. Eventually, they hit the stationary phase as they run out of food. And finally, the decline phase as toxins build up and the population dies off.
But some bacteria absolutely refuse to just die off. Pathogens like Bacillus and Clostridium form these highly resistant protective bunkers called endospores. And their secret ingredient? Calcium dipicolinate. This compound gives spores extreme heat resistance. We're talking surviving boiling water. And that is exactly why, in your hospitals, we use specifically tough spores from Bacillus stereothermophilus to test our autoclaves. If the autoclave cycle successfully kills those spores, we know for sure our surgical instruments are truly sterile.
All right, moving to our final section, clinical correlations. The pathogen rap sheet.
It's time to synthesize all this structural and physiological science into a high-yield clinical review. Notice how perfectly a bug's morphology connects right to the patient's disease. Staphylococcus aureus, forming those grape-like clusters, causes massive pyogenic infections. Streptococcus pyogenes forms chains, leading to strep throat. You see E. coli swimming around with its Paratrichus flagella? That is your #1 cause of urinary tract infections. Mycobacterium tuberculosis has that waxy acid-fast wall making it incredibly slow growing, which leads to chronic tuberculosis. Clostridium tetani forms terminal bulging spores that look like tiny drumsticks, and it releases a deadly neurotoxin. And Vibrio cholera, those comma-shaped curved rods darting around like fish in a stream, they're the culprits behind the severe rice water stool of cholera. Cement this table in your minds, guys. It's incredibly high yield.
Before we wrap up, we really must acknowledge that our adversaries are constantly upgrading their weapons. Bacteria share DNA to gain new traits, and they do this through 3 genetic mechanisms. Transformation, where they literally just pick up naked DNA from the environment. Transduction, where viruses, called bacteriophage, accidentally ferry bacterial genes around. And conjugation, where they use those sex pili we talked about to directly hand over resistance plasmids to their neighbors. Which brings us to a critical final thought for all you future clinicians. With plasmids sharing resistance genes so incredibly efficiently right under our noses, are we prepared for the post-antibiotic era? The knowledge you've gained today on bacterial structure and physiology isn't just for passing your exams, it's the very foundation you're going to use to fight back in the wards. Thank you for joining me on this explainer. Study hard, trust in the facts, and never stop questioning.